Thank you. Can you, can you hear me well?
Yes. Yeah, we can hear you.
And you can see the presentation.
And we see the slides. Yes.
Okay, very good. Thank you very much. I would also like to thank the organizers for inviting me. I would have also liked to be in Brussels, but thankfully we got sent some beer and snacks, I guess, to enjoy tonight during the beer hour. So my name is Nadine, as was already mentioned. I'm currently at the Max Planck Institute for Cell Biology and Genetics in Dresden, but in 4 weeks, I'm moving my lab to Switzerland, Lausanne, so it's busy times. And I thought, since we are such a general audience, I start with a general introduction to get everybody warmed up to the topic. Like, actually, how do I do that? Like this. So I think that one of the most exciting questions, at least to me in biology, is the question of how we get this beautiful variety of cell types that we all know of, like neurons and red blood cells, fat cells, maybe not so beautiful, but in spite of the fact that all these cells have the same genomic information. And of course, an important answer to that question lies in the differential and therefore in the differential gene expression. Here I'm showing you some beautiful gene expression patterns to illustrate this point. And so in my lab, we try to understand how the transcriptional machinery, so transcription factors and RNA polymerase, come together with the chromatin template, so the genes that need to be transcribed, in time and space to robustly regulate transcription. And to address this question, we mostly use the very beginning of transcription during animal embryogenesis. And I've depicted this here because I'm not sure how, there might be some people that don't think about embryos every day. So upon fertilization in most animal embryos and also in plants, actually, there's a period of time where there's no transcription. And development during this time is driven by maternally loaded RNA and protein, which is indicated here in red. And over time, this gets degraded until at some point, the animal takes control over its own life and starts to use its own genome to transcribe it. And so as you can see here, the time at which transcription begins in different species is quite different. And this is both true for clock time, so the number of minutes or hours after fertilization when transcription begins, but also in terms of the number of cells that is present at the time that transcription starts. But within a given species, and that's important, this onset of transcription is very precisely timed, and it therefore offers a very nice system to try and understand how transcription is actually regulated, because you go from a situation where there's absolutely no transcription to a situation where there actually is transcription. Okay. So we mostly use the zebrafish embryo to look at this beginning of transcription. And I've highlighted it here. So in the fish embryo, transcription begins at 64 cell stage for a couple of genes. And then at 1,000 cell stage, many genes are activated. So you can see that here in those two here in blue. I guess you can see my mouse. Okay. I want to end this general introduction by showing you a movie of this zebrafish embryo, which I think is always still exciting. So this here is the yolk of the fish embryo. These are 2 cells. So the embryo starts at the 2-cell stage. And I'm showing you this because I hope you can appreciate some of the characteristics of early fish development. So as you can see here, the first divisions are cleavage divisions. There's very little growth. And all of these cell cycles, so the first 10 cell cycles, happen without much transcription. This is then when transcription begins. And you can see that immediately after that, a very important event of embryogenesis takes place, which is gastrulation. So you see the cells move over the yolk and then involute. There's convergent extension. And then this movie ends 19 hours post-fertilization. And you can already see that this will, at some point be a fish. So there's a beginning of an eye here, and you can see the somites, for example. So the fish is a wonderful system for many questions, but we're interested mostly in this beginning of transcription and therefore focus on the first couple of hours of fish development. And today I thought I'd tell you 2 stories. The first one is the one that was advertised, which is about the organization of euchromatin and how we think that a process called microphase separation plays a role in this. And the second part will be much shorter. It will have only very preliminary data, but I'm excited to share some of this with you, and I'm curious to hear your input perhaps later during the breakout session. Okay, so first things first. Transcription organizes euchromatin via microphase separation. This work was spearheaded by Leonard Hilbert, who was a postdoc in my lab, shared with Vasily Zabardayev's lab at the Max Planck Institute for the Physics of Complex Systems. And he now is a professor himself at Karlsruhe Institute of Technology, also in Germany. And a lot of work was also done by two graduate students in the lab, Xenia and Tommaso. Okay, so when we started this project, we started with this very simple idea that if you look at a nucleus, which I tried to draw here, I realize it looks more like a cow actually. But so this, when you look at this nucleus from far away, we know that there's regions where chromatin is very densely packed, so chromatin rich areas, and there's regions where chromatin is quite loosely packed. And heterochromatin, which is the transcriptionally inactive part of chromatin, always resides in these chromatin-rich areas. It's heavily packed. It doesn't really need to be accessed by many factors, and therefore it can be packed away. But for euchromatin, which is the transcriptionally permissive part of chromatin, the situation is a little bit more complicated in that if it is not transcribed, it sits together with the heterochromatin in these densely packed areas, whereas if it is transcribed, you can find it in these more loosely packed regions. And together, these loosely packed regions have also been termed the active nuclear compartment because it has been shown that often we find high concentrations of RNA polymerase II there and transcription factors and splicing machinery and so on. So the machinery that actually executes transcription. And when we started this project, we were interested in understanding how you get this functional separation of euchromatin. So how do you separate it into the active and the inactive part? And so the next part of the story is about us trying to understand how you get such an organization. Okay, and here we use zebrafish blastomers, as you might have expected, but what is important is that we now don't look at the embryos when they just start to transcribe a couple of genes, because we felt that at this stage, the nuclei don't really mimic any other interface nucleus. So we took embryos at a stage when transcription is already ongoing throughout the nucleus. And we still stuck with the zebrafish embryo because it offers some important advantages. One is that at these early stages, we don't have any heterochromatin in the nuclei. And this is important because it greatly facilitates the image analysis that we wanted to do. So now we only have euchromatin and we can look at how this is organized in nuclear space. Similarly, there's no nucleoli, which also facilitates image analysis because nucleoli are quite large structures in the nucleus. And finally, these cells go through very rapid cell divisions, which is convenient because we're interested in the relationship between transcriptional activity and euchromatin organization. And so rapid cell divisions mean that we can very often look at cells as they exit mitosis and ask how the onset of transcription after mitosis relates to the organization of euchromatin. So this is for the advantages, and then there are some disadvantages too, which is that it's quite hard to image these cells as they are in the embryo because the yolk is quite big and autofluorescent. And so we actually took cells out of the embryo and cultured them as individual cells. And it's actually quite remarkable. We can take cells out of the embryo before genome activation, so before they start to use their own genome. And when we then culture an embryo and the cell that we took out of it, the genome starts to become transcriptionally active at exactly the same time. So we seem to be able to keep these cells in a very natural state. Okay, so and when we do this, we can actually use these cells for live imaging and beautiful super resolution microscopy. So we can look actually at nuclear organization at higher resolution. Okay, so let's first look at this organization and what it is that I'm talking about. So here you see STED micrographs, and we look at DNA, RNA, and the elongating form of RNA polymerase II, which for the purposes of this talk, you can just think of as transcription ongoing. And you can see that during mitosis, we can only see the DNA. We see the chromosomes, which makes sense because there's no transcription at that time. Then as cells exit mitosis, but before transcription has really started, we see that the chromatin is nicely uniformly distributed in the nucleus. When transcription has really started, so when we start to see transcriptional activity and RNA accumulate in the nucleus, you see that the chromatin domains become more grainy. So we actually see an increase in image contrast. And if we look at more nuclei and quantify this, you can indeed see that after transcription has begun, chromatin domains form as quantified by this image contrast parameter. Okay, so if we zoom into one of those nuclei, you can actually quite nicely see that in regions like this, where there's very little DNA, it's exactly in those regions that we see RNA and transcriptional activity. And again, this is not something that we see only in these large areas. That's just where we can see it very well by eye. But if we quantify it throughout the nucleus, you can see that both transcriptional activity and RNA intensity in regions where there's little DNA. And it's important to mention that it's in regions where there's little DNA because, of course, some DNA still has to be there in order for transcription to take place. And you can see that nicely when we zoom into one of those, what we like to call transcription pockets, because you can see that although there's most DNA resides outside of that pocket, there's some DNA also inside the pocket, and that's exactly where we see transcription occurring and where RNA is accumulating. So again, we can now ask where we find most of the transcriptional activity, and this localizes to regions of low DNA intensity and high RNA intensity. Okay, so from these initial images, we conclude that transcription establishes this pattern of chromatin domains that are interspersed with RNA-dense regions where transcription is localized. So this is at somewhat higher resolution, but other than that, exactly the same as what I showed you at the beginning of the talk. I want to make one more point about this. In the images that I showed you so far, we were looking at RNA that was newly made in every cell cycle. But so generally RNA, we labeled RNA with the labeled UTP. But if we look at a specific transcript, like for example here, SOX19A, you can see that the transcripts of this gene very nicely reside. They're cradled in one of the corners of this transcription pocket. And so this makes us think that these transcription pockets are made up of a series of clusters of specific RNA, SOX19A here, and gene X there, and gene Y here. So this is something that we find very interesting, but that we haven't really been able to investigate much further yet. Okay, so we see this pattern being established, and at the beginning of the talk, of course, I said what we're interested in is understanding how you get that pattern. And we now know that it is established when transcription begins, but that raises the question, what is important? Is it the accumulation of RNA, which is the product of transcription, or is it the process of transcription itself? And so in order to begin to address that question, we decided to first inhibit transcription with flavopyridol, which is an inhibitor which throws off the RNA polymerase from the genome. What is important is that we can do this in many cells at the same time. And in this case, we selected cells in which there was still RNA in the nucleus. So now we have cells in which there is chromatin and RNA, but no transcriptional activity. And I hope you can appreciate that if you compare this image here to this image, the domains of DNA seem to become more coarse-grained. So the domains of both DNA and RNA seem to become bigger. And here you can see that the DNA and RNA remain segregated like they were in the untreated condition, but the domains become larger. Okay, so I've put that also here in that schematic. If we inhibit transcription, we see a segregation of DNA and RNA still, but the domains become more coarse-grained. And so this observation led us to think that perhaps euchromatin is organized like a microemulsion. And just to remind you, generally a microemulsion consists of two phases, a hydrophobic and a hydrophilic phase, and then an amphiphile which has affinity for both of these phases. And the presence of this amphiphile prevents the collapse of those two phases into just two large compartments. So it basically allows a more fine-grained structure of the two phases that have the tendency to segregate. This is a nice idea, but then of course we had to test it, and we decided to do this by now inhibiting transcription again, but rather than using an inhibitor that throws off the RNA polymerase from the genome, we used an inhibitor that stalls the RNA polymerase. So there's no transcription anymore, but the interaction between DNA on one hand, because that's what the polymerase sits on, and RNA on the other hand, because that's the product of the RNA polymerase, remains intact, at least for a while. Oh yeah, this was a nice drawing of the amphiphile. So RNA polymerase 2, which binds to DNA and produces the RNA. Okay. So when we use this inhibitor, which doesn't throw off the polymerase, but stalls the polymerase on the DNA, we don't see this coarsening of the structure that we did see with that other inhibitor. Okay. And here you can see the quantification of this. So both image contrast and correlation length, which is another measure that we use to look at DNA structure, are increased in the presence of flavopiridol, but not in the presence of actinomycin D. Okay, so we conclude that transcriptional activity is required for the fine graining of euchromatin structure, and the function of RNA polymerase is to create an interaction between the two phases that tend to segregate. I'll get back to this in a second when we will discuss the model that we built based on this data, but I first want to show you one more experiment, and that is to look at the role of RNA itself. So now I looked at the role of transcriptional activity, but the question is, what role does RNA play? And here you can see that if we look in nuclei in which transcription has been inhibited, and we compare a nucleus where there's still RNA and a nucleus where there's no RNA, you can see that in the absence of RNA, no domain structure is formed at all. And of course, this makes sense because we saw already initially that you need the onset of transcription to actually start to see these chromatin domains. What's interesting and what we don't quite understand yet is that you need a minimal amount of RNA to increase image contrast, and at some point it doesn't matter anymore. So if you have a minimal amount of RNA, you will start to form domains, and above that, it doesn't matter how much more RNA you have in the nucleus. Okay, so this is a graphical summary of these microscopy experiments. We found that if transcription starts when cells exit mitosis, you get this very nice fine structure of chromatin domains interspersed by transcription pockets. If you inhibit transcription in a way that breaks up the interaction between DNA and RNA, this structure coarsens, and in the absence of RNA, you don't get structure to begin with. This is actually important. We did all this in these zebrafish cells that I introduced in the beginning, but we have in the meantime validated these results in mouse ES cells. And this is important for at least two reasons, I would say. One is, of course, it's nice to see that if you're interested in general principles of nuclear organization, you can see the same thing in two different cell types. But the other thing is that in the zebrafish cells that we looked at, there's no heterochromatin, which is good for image analysis. But of course, it's a weird situation because most other cells actually do have heterochromatin. And in the mouse ES cells that we analyzed, we also found heterochromatin and the same organizing principles for euchromatin, which suggests that what we see is also how euchromatin is organized in cells that have heterochromatin, which is a more abundant situation. Okay, so we did all these experiments, and then we had this idea that euchromatin is organized as a microemulsion, and we thought, let's capture this or try to capture this in a model. And the model that we built was based on two mechanisms. One is that chromatin and RNA RBP complexes segregate from one another. So this is what we saw. Chromatin and RNA tend to segregate from each other in the nucleus. I have to admit, I didn't speak about RNA binding proteins just yet, but we included them in the model for a couple of reasons. One is that in the nucleus, RNA hardly ever exists without being bound by RNA binding proteins. The second is that we know that for many other nuclear bodies, like for example, the nucleolus, RNA binding proteins are very important. And the third one, the third reason to include them is that we know that RNA binding proteins have been shown to play an important role in phase separation mechanisms. And that is, of course, what we're looking at here. Now, in order to justify including them here, we did look whether important RNA binding proteins segregate from DNA like RNA does. And here you can see an example for HNRMPA1, which is an important RNA binding protein in the zebrafish embryo. And you can see that indeed, just like RNA, it segregates from the DNA domains in the nucleus. Okay. So the second mechanism that we based our model upon features this amphiphile that I introduced to you before. And here the idea is that while DNA and RNA segregate from each other, transcribed DNA can be retained in this RNA phase as long as it is bound by RNA polymerase, because RNA polymerase has affinity for both the DNA and the RNA, and therefore it allows those two phases to locally mix with one another. Okay. But then we implemented this model in a coarse-grained lattice, and we ran simulations to see whether we could obtain the patterns that we had also seen in our microscopy results. So here we're looking again at all chromatin, at RNA, and at transcribed chromatin here as a proxy for transcriptional activity. So that's the same as what I show you for the nuclei that were exiting mitosis. And first we mimic a situation before transcription onset. And you can see that, like in the images, we observe that chromatin is quite nicely uniformly organized in the nucleus. When transcription begins, it gets more grainy. And if we inhibit transcription in a way that breaks the interaction between DNA and RNA, we get very coarse-grained patterning to euchromatin. And so this suggests that the two mechanisms that we use to build our model are sufficient to explain euchromatin organization. Okay, now finally, I want to look at how transcription pockets are set up in their first place. So we looked a lot now at static images of euchromatin organization, but we were interested in how you get one of those transcription pockets in the first place. And we first used our model to simulate a situation in which only a small region of the chromatin is allowed to become transcriptionally active. And you can see that here indicated by the orange arrow. And then RNA accumulates in the places where transcription occurs. And what you can see is that in exactly those places, DNA is being pushed away. So it looks as if transcription is taking place, RNA accumulates, and it's pushing away the non-transcribed chromatin from that exact area. The only chromatin that is retained here in this pocket is actually the chromatin that is transcriptionally active, that you can also see here in the upper panel. So the prediction of this is that transcription establishes transcription pockets by dynamically displacing the non-transcribed chromatin. And then we were lucky enough to find a situation in the zebrafish embryo where we also had isolated transcription pockets where we could look at exactly this phenomenon. So here you can see these are again zebrafish nuclei that we have imaged. These are stills from a movie in a cell cycle when transcription just begins during genome activation. Remarkably, it begins in two hotspots. I'll get back to that in the second part of the talk. But what you can see is that as soon as transcription starts here, you start to see holes in the DNA channel. So DNA seems to be pushed away from regions where transcription begins. And this live imaging strategy did not allow us to look at RNA in parallel, but when we fix these cells and then look at RNA, it accumulates exactly in those pockets where transcription is beginning, obviously. Okay. So from all this together, we conclude that transcription organizes euchromatin via microphase separation, or more specifically, as a microemulsion. The two important processes that we think underlie this are the tendency of chromatin and RNA RBP complexes to segregate from one another. And the collapse of this structure is prevented by the presence of an amphiphile, which has affinity for both of these phases. We think that the maintenance of euchromatin in such microphases, as opposed to macrophases, might facilitate access for transcription factors to specific genes when changes in gene expression need to be introduced. And finally, we think that these pockets of transcriptional activity might provide a framework for the co-regulation of genes and the formation of transcription factor hubs. What I mean by this, and it'll become clearer in a little bit, is that we now know that genes often come together when they are co-transcribed and they form hubs of transcriptional activity in the nucleus. And these might, of course, reside exactly in these transcription pockets that we have identified. Okay, so let me actually look at the time. Okay, this will be very short. Second part of the talk. I want to briefly, as I said, there's not a full story here, but just I'm excited about an observation that we made and I would like to share with you. So this is about the spatial organization of transcriptional activity, and this work is done by 3 graduate students and a postdoc in my lab. Just as a means of a brief introduction, it has become clear over the last couple of years that when you think about transcriptional activity, you should think about it occurring in specific locations in the nucleus. So there's not a uniform distribution of activity throughout the nucleus. Here, this is not our work. I'm showing you data that has been published. And here we're looking at a labeled form of RNA polymerase 2. And you can see that it localizes in these specific areas. And in some cases, it makes up very, very big clusters of transcriptional activity. And similarly, here we're looking at a Drosophila embryo where UBX, which is an important transcription factor, also seems to cluster in specific regions. Important here is that they had to actually use expansion microscopy to see these clusters because often they are very, very small. Okay, and finally, and yeah, as I already mentioned before, we also know that genes often come together when they are transcribed at the same time in a specific cell. So this has been shown by a lot of genomics approaches, but I thought I'd show a nice image, which is usually more insightful. Here you can see that there's 3 genes that they are all transcribed in this cell and they're sitting right on top of each other, which is pretty remarkable. It means that they come together to be transcribed. Okay. So this is super interesting and people are starting to think about this a lot, but it's completely unclear how such hubs of POL2 and transcription factors are formed or what they look like if you would really zoom into them, how genes would come together, do they really move through the nucleus to come to one transcription pocket to be transcribed, and finally, and potentially most importantly, how does it affect gene expression? Is it important at all? And the problem is that analysis of all these questions has been impeded by the high number of foci, they're very small size, and the fact that often we don't even know what the underlying sequences are. So it's very hard to manipulate or study these foci. And so I'm here to show you 2 foci that we have found in the zebrafish embryo that I think can help us address these questions. So here you see again the 2-cell stage embryo that you have seen in the first movie that I showed you as well. These dots here are there only for us to align the movie, so you don't have to focus on those. What we've done in this embryo is inject a fragment antibody, which is an antibody that allows us to look live at the elongating form of RNA polymerase 2 again, so active RNA polymerase 2. So play the movie. You can see that initially this localizes to nuclei, which we know is background staining because there's no transcription at this stage yet. But when transcription begins, you will see very specific signal. And remarkably, it's always occurring in 2 hotspots of activity in the nucleus. So we were really blown away by this when we saw it first. We had no idea there was such a spatial aspect to the beginning of transcription during genome activation, although we've looked at this for many years. Here you see a still of the same. So we're looking on top of the zebrafish embryo now, and now transcriptional activity is indicated in red and DNA in white. So just to say that transcription begins in 2 hotspots, and importantly, we know which gene is transcribed there. It's a microRNA, which is known to be the first gene to be transcribed during zebrafish embryogenesis, and it's actually required for the degradation of the maternally loaded RNAs that I told you before drive development in the early stages. If we remove that locus from the genome, we don't see these foci anymore. And so that's important because now we can actually study these foci. And So I would just like to propose that I think that with these very big and predictable and isolated foci, we can now address important questions about the organization of transcriptional activity in the nucleus, because it allows us to do many things that are very difficult in other systems. And just as a teaser, I'll show you one movie that we recently made that I think is really exciting. Here you see one nucleus. We've labeled nanog here in green. This is an important developmental regulator, as most of you probably know. It is also known to be required to activate this miR430 locus, so the gene that we know starts to be transcribed first. And so you can see that there's many of these foci of nanog. So apparently there's regions in the genome that allow for the clustering of this transcription factor. But then when I play the movie, you can see that some of these clusters come together, and transcription only begins once they sit on top of each other. So transcription is indicated here in purple. And we know that this is this miR430 locus. And there's many interesting aspects, I think, to this movie. One is that we can see nanoclusters, and I think the underlying genes move over microns in the nucleus to come together. And the other thing is that it seems to be a requirement for all those spots to come together to actually activate transcription. So this is something that I think will keep us busy for some years. Maybe I can talk about it some other time. For now, I'd like to end. I think thanks to the people along the way, but here are the people in my lab. Actually, the photo doesn't really correspond to the names anymore that I have here. We should make a new one. It's hard with Corona. I do want to point out some of my collaborators at the Max Planck Institute in Dresden, Jan Brugges and Alf Honigman, at the Max Planck Institute for Physics and Complex Systems, Frank Jurlicher and Vasily Zabordayev, and at Tokyo Tech, Hiroshi Kimura and Yuko Sato, who helped us with the fragment antibodies that we use for live imaging. Thank you, and I would be happy to take any questions.
Okay, thank you for this nice talk. And we have a few minutes for questions. So I see Alexandra Walczak.
Yeah. Hi, great talk. So I'm really, do you know where they form spatially, the transcriptional foci? Is it a random location, or is there some sort of, basically, as you said, there's a whole nucleus, it happens in a very precise way, and it just so happens the right gene is there. So do you know how that works?
No, that's a good question. I think, so there are two aspects to it. We know that it's always seeded by a specific gene locus. So we know that it is the transcription of a specific gene that creates these foci. What I actually forgot to say, which is important, is that we know that although those two alleles of one gene seed the focus, we know that there's also other genes coming there to be transcribed. So this is something we're interested in understanding, how those genes actually go there. Do they sense that region? But if you mean spatially in the nucleus, this we have not really looked at yet. So whether there are specific coordinates, I have no idea. I don't think I should say anything about it. But they appear so in such a time fashion that it should actually not be hard to look at this, right? Whether there's any specificity to where they form exactly.
But so basically, is there any? I mean, I guess you have some idea about something breaking the symmetry and picking some spatial position, or not?
No, I don't know. You mean in the nucleus, breaking?
Yeah, in the nucleus.
Yeah, no, I really, so all we know so far is that which gene is required for their formation. If we delete this, we don't see them at all. But where, of course, so it has to happen in the domain where chromosome 4, where this gene is located, lies. And we also know that chromosomes have their own territories. And so I guess it is in that chromosome territory. But in those early stages of embryogenesis, it's not entirely clear whether they have a very fixed position. And so it's something that we should look into in more detail. That's a very good question. Yeah.
Okay, thank you.
Okay, thank you. So there is also a question by Nimwegen. I guess it's Eric Nimwegen.
Yes, Nadine, thank you very much. That was really very exciting and stimulating talk. So I have many questions, so I'll just throw out 3 things that you decide.
Oh, 3. Okay.
What you answer. So one is anything understood at all what keeps transcription off until it goes, it starts? And Second, do you have any idea what causes RNA DNA to want to phase separate? Is it maybe an entropic effect? And third, you said, well, this may facilitate transcription factors, but isn't that sort of turning the causality around? I would think transcription factors still need to get in to start something, no?
Yeah. Okay. I'll try to answer them very quickly. The first one about the time, so why is there no transcription and then transcription begins? This is, question has fascinated biologists for many years, and we think we've provided at least part of the answer a couple of years back, where it seems to be, so in the early embryo, there's a huge amount of histones, so the material that wraps the DNA into chromatin, right? And the presence of these histones seems to not allow transcription factors to ever bind to the chromatin. So we think that over time, as the concentration of histones is reduced, transcription factors finally are able to outcompete histones for binding to DNA, and then that's when transcription begins. That raises many other questions, because of course that would suggest that genes with many transcription factor binding sites, or with transcription factors that are more potent in the competition with histones, to be first transcribed. So this is something that we're now studying, but that's a question that interests us a lot. Yes, about why DNA and RNA segregate, we think this is a size exclusion problem. So we think that the chromatin and the RNA with the RNA binding proteins just cannot mix physically with each other if they are packed in tight domains. But this is a hand wavy. I mean, I have no experimental evidence for this. And the last point is well taken. I think, of course, transcription factors, so these pockets, actually the movie that I showed you last, where you can see nano hotspots already before there is any transcription, means that transcription factors are there first, and only then transcription will begin. And so now we're actually using these two foci that I introduced at the end to understand what exactly is the sequence of events in things coming together. I think this is very hard when spots are very small, but I think we have a very good sense of which transcription factors are involved, what the gene locus is, and so now we can just label them all and ask what comes first, when is the DNA pushed away, and so on. Yeah, okay.
Okay, thank you.
Thank you.
I do not see any more questions, and we just finished on time. So now we have a break of half an hour.